Hello, students. You have stated the digestion in case of oral cavity of the human beings. Okay. You have also stated the digestion in stomach. You stated how the food material is transported from oral cavity to stomach by what the esophagus, and you stated how the digestion occurs in duodenum. If you remember my lectures, you stated that uh, carbohydrates, they are digested in the oral cavity, particularly starch, by the action of salivary amylase. Okay, but not completely. That is partially digested. You stated that the proteins are digested in stomach by the action of enzyme pepsin, and that is not completely digested. Okay, complete digestion means the the food material should be converted into the molecules which can enter into the cell membrane of the villi. Okay, I mean, the absorbable material is achieved by the digestion, complete digestion of the food material. Well, in case of duodenum, you also stated that the digestion of the proteins occurs by trypsin and chymotrypsin. You also stated the digestion of carbohydrate by salivary amylase. You stated that the emulsification of lipids occurs in duodenum, and then those emulsified lipids are converted into glycerols and fatty acids by the action of lipase. Today, we are going to discuss the complete digestion of the food material in jejunum and ileum. Okay. Well, as far as the size of jejunum is concerned, jejunum is 2.4 meter in length. Okay, and if you divide the whole length of small intestine, two-fifths of the small intestine is constituted by jejunum. Okay, and as far as the reactions are concerned, that they include what? First of all, the digestion of polypeptides. What? Digestion of polypeptides. The polypeptides, they are digested by salivary, by what? The aminopeptidase. Okay. Aminopeptidase convert the polypeptides into dipeptides. Well, the polypeptides are also digested by carboxypeptidases. Okay, and dipeptides are formed. You know very well, you stated in the structure of proteins, a peptide consists of two. Say here is, say for example, the carboxyl group. Here should be what? The amino group. Okay, amino group at the end of a polypeptide chain. If suppose these are the amino acids. Okay, if an enzyme hydrolyzes a polypeptide chain at this end, that is known as carboxypeptidase. And if a polypeptide chain is hydrolyzed here, at the amino group, and then it is known as what? Aminopeptidase. So here is the difference between aminopeptidases and carboxypeptidases. Now let us come to the next reaction. What happens in the next step? When the dipeptides are formed, as I have written here, dipeptides. These dipeptides, they are acted upon by erypsin. What? Erypsin. So here is the next reaction that is carried out by erypsin. Okay, and amino acids are formed. Ladies and gentlemen, you know that the amino acids are those molecules which are the digestion products, final products of proteins. Okay, these are absorbed into the villi, which you will study in my lecture, in the absorption of food material. Okay, so this is the final product which is formed, which is received as a result of complete digestion of proteins. Taken? Well, digestion of lipids. The digestion of lipids is carried out here in the jejunum by the action of what? Lipase. You have also studied the digestion of lipids in case of stomach. In stomach also, a small amount of what gastric lipase is produced, and that causes the digestion of lipids, but in a very minute amount. Largely, the function of stomach is to digest the proteins. Okay. Again, I will remind you that digestion of proteins here in the stomach is also partial. Okay. Now, digestion of lipids is also carried out in jejunum, and here is the third step where the digestion of the lipids occurs, and fatty acids and glycerols are formed. Fatty acids and glycerols are formed. You could see the next step. What? The digestion of what? Maltose. Maltose, you know that consists of two. That is a disaccharide. That consists of two glucose molecules. You have stated in the structure of carbohydrates. Two molecules of glucose. They form the glycosidic bond, and maltose is formed. Okay, and that maltose comes from where? Fruit. Okay, and many other food materials. This maltose is acted upon by maltase. What? Maltase. Okay, you should have stated that when a word ends with an "ase," it means what? Enzyme. Lipase, maltase, sucrase, aminopeptidase. Okay, so this is what maltase means. It is an enzyme required for the digestion of maltose. It causes the hydrolysis of maltose, and what? You can guess what should be formed. Two molecules of glucose are received by the action of maltase upon maltose. Okay. Now, there is another molecule that is another disaccharide, important disaccharide, that is found in the milk. As you check the milk, maltose is present as a carbohydrate in the milk. In many enzymes are there in the milk, and proteins are also there. Very important protein, is what? Lectin. Okay, that is also found in milk. And here is what? Lactose. that is acted upon by what? Lactose is acted upon by lactase. Lactase again is okay. Lactase, it acts upon lactose, and you stated in the structure, when you stated the structure of disaccharides, I told you people that glucose and galactose, they combine together to form what? Lactose. So here is that lactose being digested in the jejunum. In the jejunum, you see glucose plus galactose. These are received as a result of digestion of lactose. In this way, the digestion of the whole food material is achieved. The fatty acids, glycerols and galactose, these are monosaccharide, glucose, all these molecules, monosaccharides, fatty acids, amino acids, these are the digestion products of different food materials, and they are absorbed in ileum. You will study in the next lecture how the digestion, what is the mechanism of the absorption is there in case of the human beings and other mammals. Thank you very much.